Yes, great. Okay, so thanks very much. If everyone in the room who's coming in can come and take a seat, we're going to start the next session. Welcome again to everyone in the room and to all the people online. We've got 3 plenary sessions now in which we're going to, as Andrew said, report some of the science that we've been doing in the Protect team. And I really like the way that Ed and Andrew have broken down these plenaries into the various sort of factors that impact on transmission. We've got human behaviour, which we've already spoken about. We've got the environment. And then the first one, which we're about to embark on, is all about the virus. So I'm Wendy Barclay. I'm the lead of theme 5 of Protect, which uses experimental challenge studies to understand what we can about how SARS-CoV-2 transmits. My partners in crime in theme 5 are the University of Leicester, and I'm really grateful to Mike Bearer for sharing his mask technology with us to monitor the breath of infected people, and also the Animal and Plant Health Agency, who have got a lovely poster outside describing their work in theme 5, and I encourage you to go and have a look at that. But what I'm going to do in the next 10 minutes is tell you some of the science that my lab at Imperial College London have been involved with. I want to call out Jay, who's sitting there in the audience, who's produced nearly all of the data that you're going to see today. So huge thanks to Jay, and also to Maya, who's here as well, who's been in sampling people, and other people in the group who've contributed. The human challenge studies that I'll describe to you today have been pioneered and led by Chris Chu, who's my colleague at Imperial, with Ben Killingley from UCL and the Royal Free. They were funded by Bayes, and we're very grateful to the team at Bayes for making that all happen, and sort of produced, if you like, by a company called HVivo, who do this for a living. I've already said thank you to Mike, and I just also want to point out that my interest in variants and viruses is supported as well by the UKRI in a consortium of virologists known as Genotype to Phenotype, and some of the work I'll describe at the end is sort of in conjunction with that group. So respiratory virus transmission is complex. I think that message has come across today already, and people and viruses are heterogeneous. And we need to try to break it down into controllable bite-sized modules, if you like, so we can pull it apart and understand the mechanisms that underlie all that complexity. We're all different. We're people, we're the donors of the virus, but the amount of virus that we shed and when we shed it is different for a number of different reasons. Maybe we were infected by a different dose or a different route of exposure or a different virus variant, or maybe we're just physiologically different from each other and put out different amounts of droplets into the air. All of those factors are very complicated. And then the virus itself, as I've already alluded to today, the variants of the virus may differ in how fit they are in replicating in humans, how well they survive in different media like the air and environment. And then the person at the other end of that donation, if you like, the susceptible person, how susceptible are they? Are they vaccinated? What route is their exposure? For how long? What's their behavior? So all of that makes it a complicated picture. And for that reason, we employ experimental challenge models because you can control for some of those variables. So in the model I'll show you, the naive, healthy young individuals received a controlled dose, all the same strain, all the same route of infection, and all was sat in the same controlled environment. So their behavior and their environment were rather similar. What these controlled challenges also enable us to do is to study the very early events after infection, which recruiting people once they're already infected means you always miss those. So this is how controlled infection works of people. We recruit healthy volunteers, and they come into the Royal Free and sit in high exchange air rooms. And they're given a standard dose of a virus which has been manufactured under GMP conditions. And once they've been inoculated, then every day thereafter, they have lots of visits from people who take samples from them, including throat swabs and nose swabs. We ask them to wear masks to sample their breath, and we take air samples at 1 meter distance from them using this Coriolis machine. And we also take surface swabs and hand swabs to see what virus they're putting out. We read out the results in terms of the virus in two different ways. We're interested most of all in infectious viruses, and you can only see that if you do cell cultures and put viruses on plates and measure how infectious they are. But of course, sometimes that's not sensitive enough, and we can pick up the traces of virus by looking for the RNA of the virus, which we then measure with quantitative PCR. And these are the results. So basically, so far, 36 healthy naive volunteers have been inoculated purposefully with a SARS-CoV-2, and 50% of them became infected. And what's remarkable is that their shedding into their nose and throat is very similar between all of those people. They all go positive for virus on around day 2. They go positive in their throat very often before their nose. They shed most virus, though, from their nose around day 6, 7, 8, and then they clear the virus after about 2 weeks. They're no longer infectious, although we do still pick up RNA in their nose and throat swabs, which is a well-known fact that you can sort of have fragments of RNA even though you're no longer infectious. We also go in, as I say, and look at what they're breathing out, what they're in the mask, in the air, and what they're depositing into their environment. And this is a sort of summary of all 18 infected volunteers. We've never found a single positive in a mask or an air sample from somebody who was in the same sort of room but didn't get infected as defined by their swab. So these are the 18 infected volunteers. And you can see that we can pick up lots of viral RNA on different days, coinciding with when they go positive on day 2 in their swabs. And these black squares here indicate the ones that are culture positive. This is infectious virus in their masks. And you can see that we see quite high levels of infectious virus in the mask. And some people, even out to day 11 after they were first infected, are still putting infectious virus there. The Coriolis machine measures what's in the air. The efficiency of that sampling is much less. We've never, in fact, got a culture positive from that. But overall, the correlation on an individual level for one volunteer, what we measure in their mask and what we measure in the air is almost perfect. So we think that what we're seeing in the air is the product of their breath being put out. We can also find virus in the environment, in their rooms. So we find it on surfaces and on their hands. The red dots here are their hands. This is 2 volunteers I'm zooming in here. Hands are positive, surfaces are positive, and sometimes we culture infectious virus from those surfaces on their overbed tables, their bed frames, their bedside tables. What you're looking at here is that the throat and the nose PCR positivity in the shaded areas. And what I'll come to in the next slide, but is very obvious here, is that this volunteer who goes positive in their nose some considerable time after they go positive in their throat is only really putting virus out into their environment when their nose is positive. You can see that here for four volunteers. This is the one you were just looking at, but here are 3 more who all go positive in their nose sometime after positive in the throat. And in the middle here, you're looking at the mask in black and the air in red. You can see here this volunteer is only putting virus in their mask or their air when their nose is positive. This volunteer here is putting infectious virus into their mask when their nose is positive on 4 consecutive days at quite high levels. And they are also contaminating their environment, their hands and their bedside tables, etc., and sometimes with infectious virus. So I think implications is what Paul just asked us for. My interpretation of that is that the source of some of this virus that we're detecting is the nose. And maybe we might want to think about how people might treat their nose. There are nasal disinfectants that are available and being trialed, but perhaps more immediately, if you're going to ask people to wear masks, covering their nose is probably important. One of the other observations that we're very interested in is whether or not all the people put out the same amount of virus into the environment. And the answer is no. Despite the fact that the swabs are relatively homogeneous, the amount of virus that we pick up in the air and the masks is really quite different between individuals. And here I've just color-coded 4 people who are responsible for the vast majority of the viral load that we pick up in the masks and the air. So who are they? Is there anything we can understand about that? Well, of course, one obvious explanation might be that those are the people with the biggest symptoms, because some people believe that symptoms are the way that the virus is expelled. And here we've asked every day the volunteers to score their symptoms on a personal diary. And you can see that for these 3 volunteers, the virus we pick up in the mask and air does correlate pretty well with when they start to say they're having symptoms. But we have other examples. For example, these 2, here's our very high infectious shedder into their mask. They didn't score any symptoms on the day that they were putting virus out in an infectious form into the air. And here's another person who's putting infectious virus in their mask who never says that they have any symptoms at all. So pre-symptomatic and asymptomatic shedding are, I think, an important feature of this virus and make it, of course, very difficult to control. What else can we understand about those 4 or 5 people who seem to be putting the most virus out then into the environment? To try and approach that, what Jay did was develop an assay that looked at human markers in the environment and in the mask and the air, as well as the virus. So we measure for 2 different human pieces of RNA as a measure of whether people are generally emitting a lot of stuff. And I think there are 3 things to take home from this. The first is, from this plot, you can see that the infected and the uninfected volunteers in the study, of whom, remember, there are 18 of each, don't differ in how much human material they put out. So infection hasn't made them exhale or emit more stuff into the environment. If you then zoom in on the infected ones only, there isn't any difference, any pattern when they become symptomatic or not. So symptoms are not making them put out more either. And then one of the things that we're very interested in, and we're just sort of finalizing the correlations here though, is that those 4 or 5 people who we see put more virus out into the environment are also the same people who seem to put a lot of human marker into the environment. So it seems to me that the physiology of some people makes them prone to be good emitters of the virus. And if we can discover what it is about that physiology, I think that could be very mechanistically interesting, but could be useful as well for prioritizing interventions. So here's a big summary of what we found. The take-home messages are emissions are quite heterogeneous, but they're there. They're in the air, they're there in the close droplets that we capture in the mask, and they're also there on surfaces. So I'm sorry, I cannot tell you yet which one of these is the most important, but they're all there, and infectious virus is present in all 3 of those versions. I think some people are prone to be high emitters. And the nose, I think, is an important source of all of this. And you don't need symptoms. I'm going to finish up with what next. Of course, I've already alluded earlier on in the day to Delta. Delta variant appears to have emerged and is more transmissible than the original Wuhan-like virus, which caused the first wave of the pandemic. We have not yet performed challenge studies with Delta variant. Those are being planned, but we do have the capability to work with a small animal model, which is the hamster. We have not yet persuaded our hamsters to wear Mike's masks. He needs to adapt them for us. So we have another piece of equipment in which we place our hamsters and monitor what they're emitting into the air. So that's shown here. These hamsters are infected with SARS-CoV-2, and then they breathe, and their breath passes across the surface of open tissue culture plates, which allows us to measure the infectious virus which deposits at different distances from the hamster. And here's just preliminary data, lots of limitations, low confidence policymakers, N of 4. 4 animals infected with alpha variant and 4 animals infected with delta variant, but we can see infectious virus coming out from both sets of animals, but you'll see that the right-hand bars are higher than the left. The delta variant infected animals appear to be emitting more infectious virus into the air. Of course, we need to repeat, we need to delve down into the mechanism and understand what's going on here, but I think as the virus continues to evolve, this could be an important way to monitor that. So I'm going to finish with my take-home messages. Experimental infections have lots of limitations. They're not the real world, but they do allow us to understand host and viral factors that determine SARS-CoV-2 transmission as part of the much bigger picture, along with all the other things like behaviour and environment. A minority of individuals may well be emitting the majority of the virus, and some variants are definitely emitted more efficiently than others and transmitted more efficiently. And we are looking forward, we hope, to being able to continue this work with a Delta challenge and look at what goes on in vaccinated volunteers next year. So I'm going to finish there. Thank you very much for your attention. I am going to hand over to the next set of speakers in the room and invite Nicola and Susan to come up and tell you about the Imogen. And we're going to take questions for all three of these talks at the end of this session. Thank you very much.
Hello, I'm Nicola, and I'm a biosafety research scientist for the UK HSA, and I'm going to be talking about the Imagine project, which is an isolator for measuring aerosol and droplet generation. So I'm going to talk about what we've been doing important on the bacteria sort of aspects of this project. So Imagine is a flexible film isolator, which just provides sort of a controlled, clean environment, which we can use to detect respiratory aerosols and droplets. And so important, we've been just using healthy volunteers to detect their normal respiratory bacteria when they've been performing various respiratory exercises into the isolator and collecting the droplets on settle plates, which we set out on the base of the isolator, and air samplers, which we position about 1.2 meters away towards the end of the isolator. So the premise of this is we're using respiratory bacteria as a marker for other potential respiratory pathogens, and it's not a new concept. So it's been done before with cancer of airborne Streptococcus salivarius, which has been shown to be related to the risk of classroom transfer of measles. So far, we've been using the Imagine to test the efficacy of face coverings. And currently we're working on determining the risk of different respiratory behaviours. So we're doing some experiments with people talking and shouting and singing and coughing and seeing what differences we see in the droplets produced and the aerosols produced based on their respiratory bacterial counts. We're also trying to sort of determine the size distribution of the respiratory aerosols. So both of these things are currently sort of in progress for us. So this is just a summary of some of our face covering data. And the total counts are just from the settle plates. So this is just the droplet bacteria that we've recovered up to 1 meter away. And this is just a sum of all of the counts. So the healthy volunteers performed a sequence of respiratory exercises, such as a deep breath in through the nose, a deep breath in through the mouth, coughing, singing happy birthday, and counting to 100. And this is the droplets produced as a consequence of those activities. So the left, you can see the orange bar, that's no mask wearing, which is clearly higher than the others. There are a couple of outliers, which we'll kind of touch on in a minute. And the middle bar is someone wearing, well, all the participants wearing a surgical mask, and you can see that the droplets reduce to almost zero when the person's wearing a surgical mask and performing the same activities. And the final bar is wearing a face shield, and you can see that sort of falls between surgical mask and no mask. So clearly it's doing something, but isn't as effective as the surgical mask. So all of the masks that we tested have shown that face coverings are effective at reducing droplet and aerosol dispersal. But it's important to note that the level of effectiveness seems to be affected by a few different factors, such as gender, or if you're male, the presence of facial hair. And so we found that women wearing either a surgical mask or a face shield, a surgical mask or a face shield works more effectively than the equivalent man wearing those items. So this is sort of a summary of what we're doing to try and determine the size distribution of aerosol droplets. So we're using the May cascade impactor, which is comprised of 4 slides that the aerosols get deposited on. And between the slides are small jets, which increase the velocity of air, allowing the separation of particle sizes. So we expect to see the larger droplets falling on the first slide, which is approximately 20 microns and greater. And as you go down the slides, the size of particles that are landing on those slides gets smaller. So we've got some very early results based on 5 participants, but it seems, obviously we can't draw too many conclusions because it's only an N of 5, but it seems like we're not recovering much from the very smaller slides. So slide 4 is very low, and it seems like slide 2 and 3, perhaps for most people, is where we're recovering bacteria, apart from the pesky person 1 who's got a bit of a strange result for slide 1. But so hopefully we're in the process of hopefully getting some more volunteers to do this, and we'll expand our data set to see if we get any more trends. So this is just sort of a summary of the non-mask wearing data we've got. So this is from the mask study, but without none of the volunteers wearing masks. And they're performing the same respiratory exercises with a deep breath in, mouth, through the nose and mouth, coughing, counting and singing. And the graph on the left is just a total, for each participant, their total respiratory droplets. So again, up to a meter, the total sum of the counts. And so you can see that there's quite a high variation between individuals. And obviously this is only based on 15 people, but the first two are way, way above what the rest produced. And it also seems like the male population is higher dispersers than females in general. So the male is the orange bars and the green. You can see the females are towards the middle and the lower end of the counts. And so the graph on the right just shows the sum of the participants at each stage. So each bar represents all of the participants for their 10 centimeter settle plates. And you can see there's quite a strong drop off at around 0.4 and 0.3 meters from the participant projecting into the cough box. And I think it's important to know that a lot of the participants didn't disperse perhaps beyond 0.6 centimeters from where they were sitting without wearing a face mask, which obviously again highlights the variability between participants that we've seen. So now I'm going to pass on to Susie, who's been doing similar work but with infected patients. So.
Hi, I'm Susie. I'm based at the Liverpool School of Tropical Medicine, and I'm a clinical fellow with the HPRU EZI and the READ-IT groups. And as you can see, we were extremely excited to receive Imagine 2 up in Liverpool, hoping to use it with COVID-infected patients. So what we have done is we've recruited individuals who have got mild infection within the first few days of illness. So this is aiming for less than 5 days from symptom onset. And we have Imagine installed at the clinical research facility at Liverpool Royal Hospital. And whilst the patients are in there being screened for another therapeutic trial, we are able to approach them and ask them if they would mind getting involved in what can seem to be slightly unusual types of sampling for the patients, I think. So the key thing with Imagine, obviously, is this series of respiratory maneuvers, and we use the same respiratory maneuvers that Nicola has been describing. So asking them to cough, asking them to sing happy birthday, and encouraging them to do that as high a volume as possible in a room in the clinic. And we sample for a total of 10 minutes, although it usually takes them 5 or 6 minutes to complete all the respiratory maneuvers. Within Imagine, we have a Petri dish with a gelatin filter near the patient. We have the Anderson sampler, which can fractionate the particles by size. And we also have kind of an indiscriminatory Sartorius style adapter, which again is pulling air over a gelatin filter. We also measured the CO2, the temperature and the humidity within that box. In addition, we asked the patients if they can wear a wearable air sampler, which is, as you can see here, one of my colleagues is just showing kind of the relative size of it. The actual sampler is very small. The box is a little bit heavier and it's a little bit noisy. And above that, we've got the Sartorius MDA air sampler as well, which is the portable air sampler. And we use that to take samples either in the clinic room or in the household, if that's possible as well. The gelatin filters dissolve very easily in liquid media, and then we can take that forward for PCR and culture. So this is another of my colleagues who unfortunately did get infected and was happy enough to provide samples. And I should acknowledge the group that I work with at LSTM, Emily Adams' group, including Tom Edwards, Rachel Byrne. Caitlin is not involved in this project. She was just unfortunate to contract COVID, but Kate and Don Wooding as well are on the team. So far we've recruited more than 25 patients. Some of the participants have not been able to use the Imagine sampler. That's generally because they've become slightly more unwell, or there's been a reason, a good reason why not, because most patients don't find it too difficult to do the exercises that we ask of them. So 21 people have approached the Imagine, sat at it like Caitlin is doing, and performed all the exercises. That's 11 men and 10 women. The median age is 31.5 years old, and around 4 days post-influenza is on average. Because of when we've been sampling, obviously a lot of patients have been double vaccinated, but some have not, as is shown there. Now, I don't have a full set of results to present today, but I look forward to doing that in the future. This just shows kind of some examples of the kinds of things we're finding. Essentially, we're seeing a lot of variability between the patients. So some participants, almost all of the samples are positive. Some is just a few of the samples that are positive. And some we can see in the Anderson sampler that only certain particle sizes appear to be containing the viral RNA that we're detecting on the PCR. We're hoping, obviously, to take the samples forward and attempt to isolate viable virus as well, and hoping that Imagine will be able to help us do that, having concentrated all that aerosol production into such a small space. I think that's about everything. So on to the thank you slide. And I know that Nicola wants to thank Ginny, Patricia, Wilhelmina, Simon, Alan, and I've listed my supervisors there, Tom, Paul, Lance, Alan, and Jake. And obviously a big thanks to everyone who's actually volunteered to participate in the study, whether healthy or infected.
Many thanks to Susie and Nicola for that. And the final talk in this session is from Chris O'Callaghan. Pass over to you, Chris.
Thanks, Wendy. So it's fantastic to be here and listening to these talks as well. Some data I've not seen before, so it's wonderful, sort of data that you're producing. So I'm Professor of Respiratory and Pediatric Medicine at UCL in Great Ormond Street. And respiratory viruses are bread and butter for us. We see them all the time. And we know that they enter the body via the nose, the mouth, and the eyes and infect cells. And if we can prevent that, we can prevent infection. And we also know that they leave the body via the nose and the mouth as secretions or large and small aerosol droplets. There have been a number of studies done, and the studies looking at aerosol have shown RNA. In about half of the studies, you can detect that. But detecting RNA doesn't mean to say that you've got a live infectious virus there. And there are only 2 individual case reports in the world where they've taken a particle out of the air, put it on a cell, and show it can be infective. And even those, you've really got to maybe look in detail at those studies. So this makes us think that could it be our methodology? We're collecting into large volumes, and are we using the right infective cells for our challenges? So we've only come to this recently. We've only been doing this work for the last 3 or 4 months on the aerosol side. So this is very preliminary evidence. Why are we interested in this area? Well, we've got a long history in aerosol medicine. And in, I think it was 1916, we were the first to show that respiratory syncytial virus, which has been around for decades, is actually produced by young infants in large amounts in aerosol and travels up to 5 meters from the infected patient and infects ciliated epithelium. So aerosols are of interest. We're also interested in the ciliated epithelium the virus infects. And going back to 2001, we did a challenge study with coronavirus. And of interest, when we challenged them, they all got destruction of the respiratory epithelium, but 25% of them, despite this, got absolutely no symptoms. So that's of interest. So how can we develop a cell model to improve our infectivity? So we've got a model where we can go around the room here and take a brush up everybody's nose, and we can take it back to the laboratory and we can recapitulate your ciliated epithelium. This is one of our cultures after several weeks of growing. And then this is what the virus sees as it goes in. So we can put the virus on and see what happens. And we know from our work with RSV and rhinovirus that when the virus goes in, it targets the ciliated cell, and the machinery within the ciliated cell really amplifies the viral protection. And the work that we've done so far, infecting in different disease groups, has really shown us a massive spread of infectivity, even just within that respiratory epithelium. So some patients just getting 400 plaque-forming units, and some getting, sort of well over 1 million plaque-forming units. So the variability in individuals is really huge. So what we've done is try and combine these two things. So we're doing a study where we're collecting the air and fractionating it, and we're getting the people to put their fingers on coins and then looking later to see, can we actually show infectivity in the human and the Aerosols as well. Again, we've only really just started, but we've started doing home visits now. So we can actually go into the houses of children and adults with SARS-CoV-2. This is myself and our research fellow, Mira, and we can spend time with them collecting the aerosol and doing these studies. So I'm going to show you just a very few results. We've been looking initially at members of GOSH staff who are double vaccinated. And they've all got evidence on CT value. So the lower the CT value, the more likely is the viral load's greater. And we can see there's a spread. We've been looking at the skin around the face, and we see in, sort of 5 or, sort of all but 2 of the patients, we've got skin contamination. And then on the disc, so the disc 1 is immediately, so you put your finger on and we culture it, we take it immediately. Or disc 2, it's left out in the air for 30 minutes, disc 3 for an hour. And you can see we're still recovering RNA, but does that mean anything? Is it infectious? So we've gone on to look at our ciliated system. And again, this is just on one of those patients, but here we can see the cilia at the top, the blue cell nucleus, and you probably can't see it very clearly, but 3 days post-infection and 7 days post-infection, you're seeing the red of the SARS-CoV-2 spike protein on immunofluorescence in all of these, and we're extending this time course. So, and we've got an increase in RNA level as well. So we're being able to show that they're contacting, and it's remaining infectious from that patient on the surface. So again, it's very preliminary. So I don't want you, to take too much away until we can really get larger numbers. What about the aerosol study? Well, this is of interest because collecting the aerosol, we collect it into large volumes of fluid, which really negates a lot of the measurements we can do because we haven't got ways of concentrating the virus brilliantly at the moment. So again, we've been out and we've collected, and we've just got one patient where we've shown there's a CT value, and it's a high CT value. But the question is, does an absent CT value mean to say there's no infectious virus present? So this is something that we'll be doing over the next 2 or 3 months. But just to, give you a little bit of preliminary information with that patient, we've actually taken the stages, stage 3, where we got a sample positive, and stage 5, where we got a sample positive. Stage 4 was negative, but it's almost certainly positive because of, it'd be almost impossible to get virus there and there without there. And again, at 7 days post-infection, you can see the SARS-CoV spike protein. And you can see a small increase in RNA as well. So I think this gives, for the first time, a little bit of evidence that an aerosol coming out of somebody is actually containing a virus that's infectious, and it's infecting a ciliated cell. I'm not going to talk about the pediatric ones because there's not time to go through that today. But what are we doing in terms of our development? So we're trying to improve our collection of efficacy because we haven't got a machine that can really reliably collect aerosol in a concentrated form. So we're minimizing this and doing the calibrations on this, playing around with these dishes. Also, if we're going to suggest that could ciliated cell culture, and we could actually get the ciliated cell culture from the person who produces the most virus. And when we've got that, we've now developed methods where instead of, we used to get just 6 wells from each patient, we can now with our new methodology, we can get up to 300. And with BMI transformation, we can get many, many more than that. So we hopefully will have a reproducible ciliated cell line equivalent that will allow samples to be analyzed using the ciliated epithelium, because we can keep that going for much longer. So we can keep a ciliated epithelium going for up to 6 months in our laboratory. So you can look at different time periods. The other thing that we're doing in the background is we're just looking at, again, transmission. This is a patient. I don't think you can see it clearly here. This, well, a surrogate patient. And you can see that depending on what's happening in the room, this is in theatre at Great Ormond Street. If you collect the virus, if you collect your sampling somewhere else, you will miss all of this. So again, it's the positionality and the way we collect things and thinking about this. Because my biggest question to everybody I'll come up to in a second, but before that, I'll just do my acknowledgement. So our Danny Lee, Mira Santha, Colin Butler, Elaine Clampman-Green, and sort of our Aerosol team, really good collaborators at UCL and Great Ormond Street. Jay and Wendy and Ryan from Imperial have been particularly helpful, particularly early on, sort of helping us when we were developing things. And obviously Alan Bennett, Barry Atkinson, and Ginny Moore from Portland down. So is this research important that everybody's doing in this room? I think it's crucially important, because the question that really makes me worry is, if the next pandemic affected children, are we prepared? So I think it's crucial that our research doesn't stop here, and we actually try and work out what's needed. So when something comes along, we can hit the ground running. We've got our methodologies up and, sort of able to, to hit straight away. And I think that will benefit us. Thank you.
Very much, Chris. I'm assuming that's on. Yes, I'm just going to move a bit closer. Okay, so thanks very much to the other speakers for contributing to that session. I'm going to open up for questions from the floor. I don't know if there's any in the room. There's been quite a lot of chat about the implications of no's being positive. And just to sort of clear up that at the moment, my understanding is that there's a move towards no swabs only for things like lateral flow tests. I think just to clarify the implication of what I showed today, if you take it in its full implication, is that that should be okay, because we don't see people emitting virus out to their environment unless they're nose positive. So people who are throat positive only didn't seem to put anything out. Now, we can't at the moment, with the methodologies that we've done, say that they were incapable of transmitting to anybody else, but we didn't detect any virus coming out from them. So I think more is needed before one would sort of be able to say robustly that people who were not positive in the nose were infectious, but the implications are that they're not, and therefore nose only swabs are really a measure of how infectious a person is. Fliss.
I wanted to ask the ladies about the study with the visor and surgical mask and no mask. Did you do any studies, or do you have any intention to extend the studies to consider non-surgical types of face covering? So sort of different types which are readily available on the market, but which may be made out of materials such as silk, which don't have very high filtration benefits.
Yeah, so that definitely is something we're looking at. And as part of the mask study, we did a lot of work on transparent face coverings. So far, all the masks that we've tested, obviously they vary a little bit, but they do offer some reduction in droplet dispersal. But I think it is important that it seems like the fit of the mask and the wearer's face does have a really big impact on how well the masks work, which I think isn't unsurprising, really. But yeah, no, certainly we're looking at, and I think there has been some work on sort of homemade face coverings. But yeah, no, that is.
Just because it's a marginal benefit, is it better to have something which can be washed and therefore isn't going to go into the rubbish cycle?
Yeah, no, absolutely. That's something we're looking at.
Any more questions in the room? Yes, I can see one back there. Sorry, I can't tell with your mask who you are. So.
Thanks to all the speakers there. I think it's really interesting seeing that combination of evidence about the nose. And I suppose question for Wendy and maybe for Chris is have you got thoughts about the mechanism by which the nose is contaminating the environment in all the senses, both the air but also the surfaces? Is it secretions onto hands and onto surfaces? Is it sneezing into the environment, or is it breathing? And if it's breathing, how do people think that's generating aerosol in that particular case?
I'll start off and then hand to Chris. I mean, I'm pretty confident that normal breathing activities emit droplets into the air. I mean, I always sort of say when you walk down the street on a winter's day, you see that sort of cloud in front of you. So I don't think you need sneezing to push virus out from your nose. I can't exclude that sneezing doesn't enhance the amount that's output, but I think some people during their normal breathing procedure just put it out there, and some of what goes out there will have come from your nose. I think that our volunteers in the human challenge study were not really very symptomatic, and the number of sneezes reported was pretty low, and similarly coughs. The most common symptom that they had was the anosmia, and that was measured quite objectively, the loss of taste and smell. They did a testing scratch card sort of test, and you can absolutely see that coming in at about 6 and 7 days after they get infected in nearly everyone who's infected. But the other respiratory symptoms were not common and were not sort of uniform. And yet, you know, we saw plenty of emission into the air. So that would be my question to you. But Chris, do you want to follow on from that?
Yeah, I think in terms of the mechanistic point of view, you've got the ciliated cells in the nasal epithelium, and I think they're the ones that are going to be really replicating virus if it's consistent with other viruses. So you've got a reason why the nose has got huge amounts, and it has got huge amounts in. If you look at the amount of RNA in people's noses, it can be astronomical. So I think that's one of the reasons. I think if you've got so much there, then potentially by breathing in and out, you may generate some. The other maneuvers, I think, will be really interesting to look at, and how they affect the nose and the air escape through the nose. I certainly think with sneezing, you know, when you sneeze, everything comes out the nose. So you know, and you can't predict that with somebody. Sneezes just come on, and I think, you know, it's. If you're in a carriage and somebody sneezes, I'd be really worried about you. But it's a good point. I think we still don't understand fully.
Thank you.
Okay, I've got one question from Mike, and then I think back to Fliss. I am monitoring the chat. The chat is still talking a lot about lateral flow tests and noses, but I think they're having a good chat. I'm going to leave it going. Mike.
Just very quickly for Chris, you seem to imply that the ciliated epithelium was more sensitive for culture. Is that right? And do you have a direct comparison between Vero and ciliated epithelium?
So I'm not claiming that it's more sensitive, Mike. I think it's a very good model because it's actually, you know, it's the recapitulated human airway epithelium. So it's not a cell line that may react differently. So I think that's the major message. I think it will be really interesting to see if we can tease out cells from people where we do get high levels of infectivity to make our work easier, and then do a comparison with the Aerosol. So a comparison with the Aerosol is something we're doing. We did have, I didn't show the Aerosols pictures on those, but it was very difficult to see the staining at you know, sort of at the same time point. But it's so early, I don't want to draw any conclusions.
Okay, Fliss, did you have another?
Just a really short one. That statement that you made about, you know, it being difficult to tell when people sneeze. I don't think I've ever found something as guaranteed to bring on a sneeze as doing a nasal swab for a COVID test. Is there a small knock-on positive that we can start to examine the mechanics and the biology of sneezing more thoroughly by getting people to be in a room where they can be measured in certain ways while they stick a swab up their nose, because it's quite likely to bring a sneeze out?
Yeah, I mean, you can sort of try and induce sneezing. So maybe that's Wendy's next project. And there are, if you just wanted to look at individual patients, there's a tiny number of patients who sneeze all the time. So you know, there's a potential of trying to, you know, well, not all the time, but they do sneeze a lot. And the other thing is that people's sneezing seems to come in bouts. So I know I sneeze 6 times if I start off. So, you know, if you were like that, you know, sort of, at least you've got a chance to cover off over the next one.
Okay, question at the back, sorry again. masks preclude identification.
Hi, so just in terms of really interesting data in terms of what you're getting from the nose. Wendy, do you have any record of how much time people are spending vocalizing while they're wearing the mask versus just breathing?
So we didn't ask them to vocalize at all. And these are volunteers who are on their own in their room when they're asking, when they're wearing a mask. They might talk to themselves. We didn't tell them not to, but they haven't done anything other than sit in their room and breathe. So we've talked with Mike how exciting it might be for our next study to have a second mask in which they do something like sing happy birthday while they wash their hands to see how much that outputs. So I think activity will be very important to think about. Perhaps thinking about that and thinking about emissions, I wanted to go back to Chris actually and talk about children, because there's been a lot of discussion in this pandemic about the role of children. And to begin with, we felt they weren't really contributing as much to the pandemic, but I think we can now see that the rates of positivity amongst children are 5%, I think, or whatever prevalence at the moment. In your studies so far, I know you're going and looking at children and their emissions. Have you got any hint of whether or not children are emitting as much virus as adults?
No, I mean, we've really are at the start, so I wouldn't like to comment either way. We know they replicate the virus in the ciliated respiratory epithelium, so I'd be surprised if they weren't transmitting, but we don't have the evidence for that. I would think they will, but
yeah. And I suppose I wanted to turn back to Susie actually about hospitalised patients. So far, if I understand right, you've used Imagine on people who are sort of mildly symptomatic. Forgive me if I've got that wrong, but yeah, I mean, right. Again, we were approached early in the pandemic from various medical colleagues who were concerned about doing procedures, for example, tracheostomies or whatever, on patients who were SARS-CoV-2 positive. Do you have ambition to look and see, at the later, more severe end of the spectrum, what kind of emissions are coming out?
So prior to using Imagine, I've been doing sampling around 250 patients in the hospital setting who were more acutely unwell, either on AGPs or on room air or on oxygen. And we did find viral RNA in those air samples with quite a high frequency. We didn't necessarily find a lower risk of that we would detect it in rooms where patients were not on AGPs. But there's obviously lots of factors that may affect that, such as the different ventilation that's usually in play when somebody's having an AGP. So it's something that we're looking a bit more at. In terms of using Imagine with more severely unwell patients, it's a little bit more difficult for them to comply with the singing and so on.
Sure.
Yeah.
Yeah. Okay. Any further questions or comments in the room? I'm just going to take a final look down on so there's been lots of stuff in the chat. I don't know if everybody else is going to go and look at this, but basically lots of track and trace conversations about children and PCR test dates, etc., and how long people should be positive. I mean, what we hope with our human challenge studies is that we are providing some science evidence base for this sort of issue of how long a person should isolate for, etc. But again, limitations and confidence. We've inoculated people with one dose through one route, and they're healthy volunteers. We've done that in order to try and remove some of the variables. But overall, there could be other variables that we don't control for or don't know about in that experiment, and that is the limitation of those experimental challenges. So, at the end of the day, the point of this study, I think, is to gather experimental evidence from as many different real world and experimental situations as possible and try and inform policy by those procedures. And perhaps that's a place to end, really. Thanks very much, everybody, for coming to this session. And I think it's lunchtime. Great. Thank you.